Good day, everyone. My name is Estrella Villamizar. I'm a researcher at the Central University of Venezuela, and today I'm going to talk about our research findings on the biodiversity in coralline bottoms of the Archipiélago Los Roques National Park. Our objectives are: 1, to increase knowledge about the biodiversity associated to coral communities in this marine park, and 2, to establish a biodiversity baseline to contribute with the management and preservation policies of this unique coral reef system in the Caribbean. The study area. The archipelago is located about 157 kilometers from Venezuela's mainland, north of the central coast. It has 2 large barrier reefs, one to the south and one to the east. The yellow and green circles indicate the survey sites. Yellow sites are the most frequently sampled, and the yellow star indicates the location of the marine biological station. For benthos and fishes, we apply the AGRA protocol. We also perform visual census to include the species from different reef bottoms. Results. A total of 9 phylum were studied. From these, 6 were identified at the species level. In this pie chart, we present the number of species of the 3 main components of the system: plankton, nekton, and benthos. Out of total of 1,700 quantified species, almost 65% is represented by benthos species. In this figure, we show the contribution of each phylum to the total benthos diversity. Numbers above bars indicates the expertise and research effort from the particular phylum. The mollusks, macroalgae, and crustacea are the taxa that contribute the most to benthos diversity. These results could represent 1, actual differences in taxon diversity, and or 2, uneven sampling effort between groups, depending on the number of researchers for each group. These next 2 slides contain pictures from the different studied taxa. Numbers point out the richness of species on each group. For example, this is for corals. We found a high spatial variability in the coral species richness along the archipelago. The graphic shows the number of species for the 28 study sites. We found that more species richness is found predominantly in the most frequently sampled site: Cayo Sal, Dos Mosquices Sur, Boca de Cote, and La Pelona. Diversity analysis. Considering that much of the richness of organisms on the reef depend on the diversity of corals, sponges, and gorgonians, we estimated the alpha and beta diversity of these groups for 1, the 5 more frequently sampled reef, pulling the data, and 2, only for Dos Mosquices Sur reef. In these tables, we show the results. In both cases, alpha diversity was largest for coral communities and beta diversity for sponge communities. The magnitude of both indices was higher for the most frequent sample sites. Conclusions. The National Park Archipelago Los Roques is a Venezuelan coral reef system of very high diversity of taxa. 80 new registers of species for the archipelago and 27 for the country had been added to this date. Second, from the studied taxa, fishes, mollusks, crustaceans, and macroalgae have the higher number of species. However, other groups not studied yet, but with a very limited sampling effort, could overpass the richness of these groups. Third, the estimation of species richness of this reef system depends on 2 main factors: the geographical scale of the surveys within the archipelago and the sampling effort. Also, the type of coral formation must be considered. Finally, the diversity indexes indicate that coral communities have the largest alpha diversity, and sponge communities have the largest beta diversity or turnover rate. Acknowledgement to all researchers involved in this project, and also to sponsors, and special to Fundación Scientific Los Roques, members and personnel of the Mosquices Marine Biological Station. Thank you.